who were expecting something else, I'm going to be talking about the five senses, and hopefully, or maybe if I survive this talk, I'll have enough energy to do part two. And the very last slide or two that I have in this group is actually sort of an introduction to what I think part two is going to be about. But this part's going to be about the five senses. And everybody knows everything about the five senses. You grew up with them in your science textbooks starting in grade school. And there's nothing new to learn. And hopefully I can dissuade you of that. Next slide, Lou.
Okay, there we go.
All right, so everybody knows that we have five senses at least, and sight, hearing, smell, taste, touch, and they're how we perceive the external world, which is called exteroception. And we all think we know the organs of the five senses. And early information about how all of these senses worked came from basically gross anatomy, dissecting people. Later, we got a lot of information from research on defects like what causes color blindness, or from battle wounds in particular that ended up cutting nerves and therefore stopped certain parts of our senses from working. But I'm going to focus on the last 20 to 25 years, where we've been looking at the five senses from a chemistry and a molecular biology point of view. And so hopefully that's going to be the new part that you haven't seen before. And I'll try to keep it non-technical, but it's hard to give a technical talk and not give a little technical information as part of it. Anyhow, next slide, please. All right, so all of these senses work in basically the same way. The organ of the sense contains specialized tissues. Those tissues contain specialized cells. Those cells contain sensory receptors. So, for example, for sight, they're photoreceptors that interact with light. For hearing, they're mechanical receptors. For smell and taste, they're chemical receptors. And for touch, there are three different kinds. They're all considered neuroreceptors. Some of them work mechanically. Some of them work in ways we really don't understand. And some of them are basically nerve endings with specialized gadgets at the end of the nerves that react to various aspects of touch. Next slide. All of these sensory receptors act in a similar way. A specific stimulus, like light, activates the specific type of receptor. The activated receptor generates an electrical signal. That electrical signal travels via specialized nerves to the brain, and the brain interprets the signals it receives. And if you look at the little diagram at the bottom, you can see that in simplified form, each of the senses has its own part of the brain that's the initial contact with the signal from the organ via that particular kind of signal generating receptor. Next slide. So all of them work the same way. And I'm going to start with the eye for a couple of reasons. One, because it's something that everybody thinks they understand pretty well, at least in gross terms. And two, because I was in on the ground floor of the chemistry that is actually at the heart of why sight actually works. So, eye is the organ of sight, and at the back of the eye is that thin membrane, and that's the tissue of sight. Next slide. Laid out on that tissue are these strange little long gizmos shown on the left panel, and I'm pointing at my screen even though you can't see it as usual. But those are the rods and cones, which are the cells of sight. And as you can see in the right panel, the rods are pretty much symmetrically distributed along the entire retina in a normal distribution, except there's a big hole right in the middle. And that's where all the cones are. And there's a little bit of overlap, you know, and the angular separation. The fovea is the central dark line down the center of the retina in the diagram. And so at 20 degrees from either side, the cones peter out, and that's where the rods really start taking over. Now, there's a lot of rods, 120 million of them approximately in a normal eye, and only 7 million cones. And there's a reason for that. Next slide. And it's because the rods have the responsibility for, hang on, I'm having trouble seeing my own slides, so I've got to get rid of something. There we go. The rods are very sensitive to white light. And as you look over on the right side, it's only the rods that are functional in low light and are responsible for night vision. And in order to have the sensitivity to see where we're going in the dark, basically, we need a lot of rods. The cones, on the other hand, take over during the day because it's that sensitivity of the rods that also makes them useless if the light's too bright. And so
exposure.
Daylight vision is mostly cones and night vision is mostly rods. And there are three kinds of cones. Ones that are sensitive to short, medium, and long wavelengths. There's a lot of overlap, especially between the medium and the long. And they provide image sharpness and color information as well as just overall light sensitivity. The other thing that you can see from the diagram on the right is that the efficacy, and luminous efficacy is a terrible term. I wish I had been able to find a similar diagram with a different ordinate scale, because nobody, including me, really understands luminous efficacy completely, I think. But the point is that rods are too sensitive, as I said, and cones are not very sensitive. So they die when it's low light and the rods take over. And the other thing you can see is where the peak is. The rods have a peak in the blue-green. And if you remember seeing the movies where they're looking through night vision scopes, you remember what those colors are? Everything is in sort of a bluish green type of tone. And that's why, because when you're light gathering, that's the wavelength where the rods are most sensitive. Next slide. All right, so now the rods and cones are the cells. All of them contain a chemical called cis-retinal, which is that long-looking thing over on the left side, the molecule. On the right side is transretinal, and if you look at where the red arrows point, you can see that the only difference between those molecules is the shape of the molecule around that double line. That's called double bond, and it is that simple change in the shape of the molecule, not in the formula, not in any other aspect of the molecule, just in that simple shape that's responsible for sight. That particular reaction is called an isomerization. In the case of cis-retinal, which is a chromophore, it absorbs visible light. And so that's why we see in the visible spectrum. I got involved with vision, as I said, because I was in on the ground floor. That particular photochemical reaction was being studied when I was an undergraduate at Caltech, and I did undergraduate research in George Hammond's lab, and he's basically the father of organic photochemistry. Used to be the job of physicists to look at photo reactions, but George showed that they were important in organic chemistry as well. And I got to work with all of the grad students who were just starting to work on that particular reaction, including a fellow named Peter Learmakers. And when Peter Learmakers got his PhD and eventually became a professor at Wesleyan University, one of his research topics built on that simple reaction that he had studied as a grad student. And he started to look at how that reaction happened in the eye itself. And it turns out that cis-retinal ends up being bound to a protein called opsin when we're talking about rods, and that produces a pigment called rhodopsin. Now, the cones use 3 slightly different binding proteins and produce 3 slightly different rhodopsin equivalents. But that's really not very important for this discussion. An interesting aside is most of you, I'm sure, have heard your mother say, "Eat your carrots or you'll go blind." Well, it's true. It turns out that the source of cis-retinal that we use in this reaction comes from vitamin A, which is a rich vitamin in carrots, and a couple of simple chemical reactions, enzyme type reactions, ends up turning retinol, vitamin A, into cis-retinal, which is the basis for this photochemical reaction. Next slide, please. So how does this work in the eye? So at the bottom left is a rod, and you can see it looks like a rod. It's very long. And the top part of it has a stack of discs. That's all one cell from top to bottom. And each of the discs is shown in the upper left. It's the rhodopsin molecule with the cis-retinal in the center of it. And it's bound on both sides by a membrane to keep it all in one place. And so the top of the rods that are facing to the light as it comes into the eye is this stack of discs. And the reason that the rods are so sensitive is one disc undergoing the reaction from cis-retinal to trans-retinal is enough to send a signal down to the end of the rod and into the optic nerve. And a whole bunch of them will do the same thing because the first photon gets trapped by the top disc, and the photons keep going, and eventually the rod gets deactivated if there are too many photons because all of the discs have reacted, and it takes a while for them to recover. And so that's another aspect of this. We can understand why, if we're exposed to too much light, we essentially go blind for a while and we can't see anything. It's because our rods and also the cones work in pretty much the same way. Our rods and our cones have been saturated and they need a chance to recover before we can see again. Okay, so what happens? We've got the rhodopsin and the opsin binding protein. Because of this cis-trans isomerization with light, the opsin binding protein changes shape. And that makes the bathorodopsin, what it turns into, unstable. And that bathorodopsin starts undergoing all sorts of what are called conformational changes. The shape changes in a whole series of steps until eventually we produce a compound called metarodopsin 2. And obviously there's a 1 and there's a whole bunch of others. And even metarodopsin isn't completely stable, which is why it's called meta. It's transitory. But metarodopsin, which is in this stack of discs at the top of the rod, then undergoes a 4-step signal transduction cascade. And all that means is the energy that was collected by all of the rhodopsin molecules in the rod get transferred into an electrical signal, and that ends up going down to the bottom of the rod. And the bottom of the rod is a synaptic junction with the optic nerve, and the signals that are produced transfer across that synaptic junction. Next slide, please. I promise this is the only scary one I'm going to show. Just to prove that molecular biology is important in all of this, this is the signal transduction process in 4 steps. And so you can see at the left over there that light's coming in and activates the opsin molecule. The important thing is that what it activates is something called a G protein, in this case, transducin. There are lots and lots of G proteins, but they all serve the same function. They act as molecular switches inside of cells by opening and closing various kinds of ion channels. And it's those ion channels which actually produce the electrical signal that ends up going to the nerves. In this particular case, the ion channel is that red thing towards the right. That happens to be a very unusual kind of ion channel, because instead of being controlled by an ion, it still lets ions pass through or closes, but it's controlled by a cyclic nucleotide called cyclic GMP. And in this case, the other unusual thing about this particular process is instead of producing a positive signal, this process produces a negative signal. Because what happens after all of these steps is that the ion channel closes and stops the release of glutamate, which is the chemical that goes across the synapse and fires the nerve. And so the 4th step is over on the right, and it's basically a restorative step that synthesizes cyclic GMP and opens that C and G channel and releases the glutamate. So in this case, the signal acts backwards. The signal is constantly on, and suddenly it gets shut off and then on again. Next slide. Okay, so what happens to the signal? And this is a very, 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 very simplified version, because I didn't want to take the whole hour talking about vision. Basically, what happens is the nerves come from the eyes, they go down the optic canal, and one of the things to see is that there's not just one nerve. The optic nerve is actually a bundle of nerves, and it turns out that that bundle of nerves, even though they all go to the same place, that bundle of nerves has spatial information associated with it, because each of those individual nerves comes from a different part of the retina. So from the outsides of the retina and then towards the center. So the cones have their own nerve, and various groups of rods going further and further out along the retina have their own nerves. And so one of the things that happens is the raw signal provides information to the brain as far as the geometry, if you wish, of the object that we're looking at. And that's an interesting aspect of all of our senses. The signals undergo a lot of pre-processing before they ever get to the brain. The mechanics or the chemistry of the process that produces the signal very often refines that signal so the brain doesn't have to do all of the heavy lifting. So I've left a couple of things out here. The signal travels along the optic nerve, and its first stop is to the thalamus. And the thalamus is like a gatekeeper for all of these different kinds of nerves from all over the body. And it sort of parcels out nerve impulses to the various parts of the brain, especially the higher functioning parts of the brain. So in this case, there's an intermediate step where the signal goes through the thalamus and then goes to the primary visual cortex. And that's that little itty bitty part in the back of the brain. And if we look at it in a back view, it's actually not that small, and it's actually pretty complicated. It has 6 layers that are functionally distinct as far as their signal processing jobs. And so there are parts that are associated with shape and color and spatial organization and movement. On top of that, the primary visual cortex parses out certain parts of the jobs to even higher functioning parts of the cerebral cortex. In particular, and I got to look at my notes to be sure, the temporal lobe, which is responsible for object recognition or helps in that process, and the parietal lobe, which is responsible for object location. It helps us grab something. It tells us where in 3D that object is, not just what it looks like. The other thing to understand about how all of this works is that all of this processing is based on differences in light intensity, the strength of the signal, how many of those discs get fired in a rod, and where the rod is along the retinal display. So all of that processing is just based on differences in light intensity, and the brain organizes all of those signals to make sense of it. And then it integrates that information using past experience to actually form the image in the brain that we think we're looking at. But all we're really looking at is a bunch of signals that started out with that simple chemical transformation. Next slide. All right, now the ear is another one of these senses that everybody understands. And I thought the same thing until I started actually looking at it. And I got as far as knowing what the cochlea is, and then all hell broke loose and I didn't understand the thing without a lot of additional research. But let's give it a try. So the first thing that happens is a sound wave, which is really a pressure wave, a vibration in the air, if you wish, is collected by the outer ear, and the shape of the ear canal actually amplifies that sound wave by the time it reaches the end of the ear canal and impacts the eardrum. The eardrum transfers the vibrations from that sound wave through the bones of the middle ear to the cochlea. And there's those 3 little bones that most people are familiar with. And so the question is, why the hell are they there? And the answer is the movement of those bones when they're affected by vibration is fulcrum-like. And that fulcrum-like process produces a fourfold amplification in the pressure of that sound wave. So now we have a second amplification step, and it's all mechanical so far. The cochlea itself is actually a very thin spiral of bone, and it really does have about 2.5 turns. That's the actual shape, but the bone is so thin of the cochlea itself that an extension of the temple bone, the temporal bone, grows around it and protects it. And so if you get rid of the innards of the cochlea, what you're left with is a temporal bone that shows the same shape in pretty much 3 dimensions. So it's not a flat spiral, it's a spiral that has 3 dimensions to it. Now, one of the reasons that it has this funny shape is because the cochlea, as we're going to see in the next slide, next slide, please. There we go. Is about 3.5 inches long. And if it didn't wind around on itself like that, there wouldn't be room in our heads for it. And so it's pretty much a function of evolution. We want a long cochlea, but we don't have room for it in our head, or animals don't have room for it in their heads. And so it winds in on itself in order to provide the length that's required. Now, the cochlea is pretty complicated, and if we look at the right cross-section first, we see that it's got 3 channels through it. The 2 outside channels are connected through a little hole called a heliocentrema, which is shown on the left panel, and they're filled with a fluid called perilymph. The cochlear channel, the one in that triangle-shaped center, is filled with a different kind of fluid. And it's very important that that fluid has a high concentration of potassium ions in it, and that makes it conductive. All right. So what happens? The vibrations in the stapes, now we're going to go over to the far left, and there's our little stirrup. The vibrations in the stapes transfer to the membrane at the start of the vestibular canal or duct. And that creates another amplification step, because the eardrum, the tympanic membrane, is much bigger than the oval window, which is at the start of that duct. And that size ratio gives us another approximately 10-fold amplification in the pressure, because it's needed as waves going through bone or air suddenly get transferred into liquid and become pressure waves. And if we didn't have that extra pressure, they'd just damp out and disappear. Now it gets more complicated because the pressure wave in the vestibular duct, the top thing, gets transferred through the vestibular or Reissner's membrane into the cochlear duct. And there's a reason for that, as we'll see on the next slide. Next slide, Lou. All right. Those vibrations in the cochlear duct become traveling pressure waves. And they look pretty simple in the top of the illustration on the left, but they're really very complicated because the cochlear duct has a funny shape. and changes in width as we go from one end to the other. But the important thing is that the frequency of that traveling wave is still exactly the same frequency as the sound that reached the outer ear. So the frequency has been preserved in all of these different transformations, from air to bone, now to liquid, to a second kind of liquid. Now the basilar membrane runs along the bottom of the cochlear duct, and it's got a very funny shape. It's very thick and narrow and bound very tightly by the cochlear duct when it's next to the stapes, but the other end of it is loose and it's very thin and very wide. And that shape is very important because the basilar membrane acts like a sounding board, and the traveling wave causes the basilar membrane to vibrate as it passes. Now, each point along the basilar membrane vibrates at the same frequency as the traveling wave, but because of its shape, there's a maximum distortion at a particular point along the basilar membrane for each particular frequency. And so if you look at the picture on the far right, what you see is the basilar membrane is sort of like a big tuning fork. And the very high frequencies that have a lot of energy can get it to vibrate at the very thick, tightly held end, but it doesn't take much energy for the very low frequencies to vibrate the basilar membrane at the very end of the cochlear duct. And you can see all of the different frequencies that go around and down the cochlea as the energy is dissipated, and you can see the wiggle, which is the basilar membrane vibrating by a particular frequency. All right, so far, no chemistry, no molecular biology. Next slide. Now, finally, we get to the business end of all of this. On top of the basilar membrane is something called the organ of Corti. It's that little red thing in the center of the illustration on the left. And it is not an organ, it's the tissue of hearing. All right, in that tissue are cells, and they are the cells of hearing. And there's two different kinds, and there are lots of them. But you got to remember that all of these hairs are spread out along 3.5 inches of the basilar membrane. And at the top of each of the hairs, and you can see an actual photomicrograph of those hairs in the center, at the top of each of those hairs is a little group of what look like hairs, but actually are membranes protruding from the top of cells, individual cells and these are called stereocilia. And the stereocilia are the mechanical receptors of hearing. So hearing is basically a mechanical process all the way down to almost the very end. So as the passing pressure wave distorts a small area of the basilar membrane, it presses the stereocilia into the membrane, the tectorial membrane above it. That distortion bends the hairs in that group, and that bending generates the electrical signal that turns into hearing. And there's lots and lots of hairs along that 3.5 inches of basilar membrane in the organ of Corti. And so again, you get this fantastic amount of signal amplification just from the mechanics of hearing. Next slide. Now, there is a picture of the cell that contains the stereocilia on the left. And you can see that the stereocilia are really extensions of the cell membrane that stick out into the fluid in the cochlear duct. So the pressure wave bends the stereocilia. A potassium channel opens. Remember, this is a potassium-rich fluid, and this is why. The potassium channel opens, potassium floods into the cell, the membrane depolarizes, that opens another channel, and calcium flows into the cell because of the open channel. That calcium then reacts with the synaptic vesicles that contain the chemical that's going to go across the nerve junction, and the signal is released. All right, so that signal travels through the auditory nerve, or really it's called the cochlear nerve, via the thalamus to the brain's auditory cortex. And this is a really complicated part of the system. It's that pink, green, and orange thing in the center. So it's got three different parts. And those parts integrate and start the interpretation process from those signals from the receptor hairs. Now, the frequency sorting has already been done in the cochlea, and so that information is retained in the auditory cortex. But the auditory cortex then performs other basic sound functions. For volume, it interprets how strong the signal is. The tempo, it interprets how often certain frequencies impact the ear. And the pitch, it determines the relative frequency, high or low, versus the rest of the signals. And then the primary cortex acts as a traffic center and coordinates with other areas like the blue and yellow one. And those are suddenly higher centers in the brain, and they start providing our understanding of music, our understanding of speech, and they even provide echolocation, if you wish, where sound is coming from. And so that provides our stereo sound capability. And then the AC stimulates even more areas of the brain, the speech center and the hypothalamus, which is the emotion center. And you can see now, because of the connection, why music can produce emotions in most people. Because there's a direct connection, if you wish, through the auditory cortex. Now, I've sort of done a hand-waving description of all of these processes because they're very complicated and we really don't understand how they work. We just know that different parts of the brain are involved in different functions. So we know even less about how the brain coordinates all of this information than we do for vision. Next slide. This is hopefully a little bit easier sense to talk about. But again, I was intrigued by this one because the mechanism of smell was only understood in 1991. And one of the researchers who discovered the process and how it works was at UW. And she won the Nobel Prize in Medicine in 2004. And I was at UW shortly after that and had the rare opportunity to hear her repeat that Nobel Prize lecture to the med school at UW. And I was basically blown away by that lecture. It was one of the best lectures for a relative lay audience that I had ever heard. And you'll see why I was so blown away when you see how the organ of smell really works. All right, so we breathe in air. It goes through our nose. There are 3 nasal passages, and at the very top, the smallest and longest nasal passage, there is a mucus layer, and that mucus layer absorbs odor-causing substances. Now, in that mucus layer and in that epithelium that lines that upper nasal cavity, there's a very, very tiny area, which is about 1/3 of a square inch in humans, and we've got 1 in each of our nostrils. But that's the tissue of smell. All the rest of the nose doesn't matter. It's just a collection device, sort of like the outer part of the ear. And in that little tiny area, there are millions of receptor cells. And in this case, they're chemoreceptors. They're very funny cells because they're very long and skinny, and they project through both the top and the bottom of that epithelial layer. The bottoms of the cells have little hairs, cilia, that extend into the mucus layer and collect odorants, the substances causing our smells. And the tops of the cells are connected to our part of the tops of the cells are nerves that connect to something called the olfactory bulb, which is protected by a layer of bone called the cribriform plate. And so that illustration on the right is actually true. All of those nerves from all of those receptor cells go through little holes in the cribriform plate. And the cell is wrong because the olfactory bulb is really not the organ of smell. It looks like it might be, but it's not at all. Next slide, please. So, what I've shown on the left is the smell chemoreceptor. And the surprising thing is that there are only about 450 different ones in humans. But there's tens of thousands of each type in that little patch of epithelium. And they're very unusual in the sense that the cells are basically their own G receptor or G proteins. And so there's almost a continuum of odorant capture by the chemoreceptor and signal reduction. So a very, or signal transduction. So a very, very fast process. Now, the other thing that's unusual about these smell chemoreceptors is that the receptor, the business end that the red arrow is pointing to, it's not specific for one substance like most receptors are. We want most receptors in other processes to be one-to-one specific, key and lock kind of thing like you've heard about for enzymes. So these receptors bind to many similar substances. And so, for example, octanol and heptanol, straight chain alcohols, one with 8 carbons in it, the other one with 7. And it binds to those two with slightly different affinities. And so what we have is this huge network in this little tiny patch of these 450 different kinds of things that are capturing odor molecules. What happens is, as I said, the capture cell, which is sort of a self-contained unit, generates a signal that travels along the nerve protruding from its top. And those individual nerves are axons from the same type of chemoreceptor. So we've got about 450 different types. Each of the types, which is spread out across that little area, all of the nerves from their tops collect in, after they've gone through the hole in the cribriform plate, they collect in the olfactory bulb, and they collect in a tangle of nerves called the glomerulus. And the olfactory bulb in humans contains about 5,000 of these nerve tangles at the very bottom of the olfactory bulb. And about 16 of the nerves of the same kind lead to each of those glomeruli. And so the first level of signal processing actually occurs in that little glob of nerves. And it provides signal amplification because of the fact that it's got this big net collecting odors of a similar kind. And it actually provides cancellation of background odors, because every place smells and we're looking for one specific one. Next slide, please. Now, the next level of signal processing occurs in the olfactory bulb, which has layers, as you can see in the left diagram. But the key layer is the one that contains the mitral cells. Those are nerve cells. And the glomeruli pass the signals after they've been processed to those mitral cells. And then there's two other kinds of cells, tufted and granule cells, and they basically provide stimuli and repression, and you get feedback loops that help refine and stabilize the signals received from the glomeruli even more. And it's that secondary processing that basically provides all of the odor discrimination. And you end up with a signal for an odorant as a result of all of the signal processing that happens in the olfactory bulb. The signals then go along the olfactory tract from all of the different smells that have been collected. And unlike the signals from the brain or the signals from the ear, they don't go through the thalamus. They go directly to the brain's cerebral cortex and to three parts of it. They go to the hippocampus, where the odor is stored in memory. They go to the amygdala, which is responsible for unconscious emotional responses. And that's why certain smells cause anxiety or fear, for example. And they go to a third part, which is sort of funny, the reticular formation, which is responsible for involuntary responses, visceral responses. And so if you want to know why the smell of vomit causes other people to vomit, that's why. If the smell of vomit reaches the reticular formation, it causes us to vomit, and that's an involuntary response. The signal then returns to the thalamus. It bypassed it the first time, but it comes back to the thalamus and to the hypothalamus, where conscious responses are controlled. And so now we have all of the conscious responses. I smell bread. I want bread. Where's the bakery? I'm going to the bakery. That kind of thing. That's a conscious response. It's the same signal, but it's gone to the hypothalamus. And the hypothalamus is where those kinds of conscious decisions are made. Next slide. We're getting through. Okay, now the tongue and the organ of taste. Now, in spite of the fact that the taste is probably the most primitive of our senses, we still don't understand it. Part of the reason is that a German scientist named Hannig did us a great disservice. He went to Japan and published something called a tongue map based on very, very shoddy experiments. And he said there are 4 basic tastes, and the tongue is divided up into parts, and each of those parts is responsible for tasting a particular one of those basic tastes. So even though he was in Japan, he ignored the work of a scientist named Takeda, who had defined the 5th taste called savory. And it was scientifically recognized in 1908, but because of Hanig and his work, little research was done on how that particular taste works until the 1980s. And it really wasn't until the mid-70s that this whole concept of a tongue map and the simplicity of understanding that it provided was debunked. And so the real research got started. So let's see how taste actually works. As most of us know, there are all sorts of little bumps on our tongue. There are various sizes and there are various amounts. Those are called papillae. And there's 4 different kinds: vallate, foliate, fungiform, and filiform. And each of them has a different amount of numbers and different taste buds. And I'll show you what a taste bud is in a second. But you can see in the picture, and we'll look at the vallate, for example, it's got 2 grooves on the side of that big lump in the middle. And there are 2 nerves that feed that lump in the middle, and there are little bumps lined up on the membrane side of those grooves. So those grooves trap taste molecules and present them to the chemoreceptors that are in the taste buds. And that's how they work. And so each of these has a different number of taste buds and a different number of nerves, but they all have the grooves with the bumps on the side, etc., except for the filiform, which are the major kinds of bumps, the major kinds of papilla. And they don't contain any taste buds, so they don't really taste anything. They're actually touch sensors. And we'll see why that's important in a minute. Next slide. All right, so what's a taste bud? The taste bud is what contains the chemoreceptors. So the real question is, where were the tissues? We know where the cell of taste is. Where is the tissue of taste? It's not the tongue, that's the organ of taste. So it's the layer of skin along the surface of the tongue, and it's the epidermal tops of all of those papillae bumps, and those are the tissues, and there's different kinds, which is one of the complications of taste and our understanding of it. So let's get back to the basics. There's supposedly only 5 basic tastes that we can detect: sweet, sour, salty, bitter, and savory. And they're detected by 3 basic types of chemoreceptors. The sour one is simple. It's a type 3, and it detects basically acidity. So it's a pH meter. And it's also got a slight amount of sensitivity to sodium ions. But that's what gives us the sour taste. The salty taste is pretty simple, and it's the flip side. It's a type 1 detector, and it detects sodium ions, but it also detects other alkali metals like potassium ion to a lesser extent. And that's why we can substitute potassium chloride for sodium chloride to give us a salt-free diet and still have the taste of salt in our foods if we need it. And finally, there are the type 2 detectors, and there's 3 types. They all are involved in the other 3 tastes. Umami is a simple one. It's got a G protein as a receptor, so a very complicated one, while the other two receptors are actually pretty simple. I didn't even bother to show them. And it detects glutamate, as in monosodium glutamate. And that's why the Japanese found it first, because they use glutamate on everything. But it also detects other L-amino acids and even small proteins. Then we have the sweet taste. It's also a type 2, but we really didn't know how that worked until at least 2002, when we got some idea of what the receptor looked like. It's another one of these very complicated G proteins. And surprisingly, sweet is the least sensitive of all of these fundamental tastes. We need lots and lots of sugar in solution in order to tell it's there. And that's one of the reasons, for example, that soft drinks need to be loaded up with this stuff in order for us to know that it's sweet. Now, that particular receptor detects not just sucrose, which is basically glucose and fructose, but also lactose and aldehydes and ketones and sugar alcohols. And the reason for that is that there are over 25 different subtypes of this particular G protein that are found in humans. And it's the business end of the G protein, that receptor end that I showed you in a simplified G protein earlier, that creates all of these different subtypes. And these different subtypes bind different sugars in that extracellular domain. And finally, there's the bitter, which is literally the most sensitive of all of the basic tastes. We can detect 1 part in 2 million of quinine in solution, which is pretty remarkable. And again, it's one of the Johnny-come-latelys. The receptor was only characterized in 2002. And that's because it's even more complicated than the G proteins for sweet and umami. It's this thing that's illustrated on the bottom. It's called a 7TM because it's got 7 parts to it, all in sequence in those little round cans. And it's got highly conserved regions in those cans. And then it's got all of these protuberances that stick out, including the one that's labeled N, which is the business end, the receptor part. And all of the red parts are variable. They can change in that family of 7TM receptors. So there are actually over 40 different subtypes found in humans. And so suddenly we've gone from 5 basic tastes to at least probably close to 70 basic tastes because of all of these subtypes of the receptors. And we didn't know any of that until we started looking at the molecular biology behind all of these taste sensations. All right, I sort of skipped what a taste bud is. So a taste bud is this spherical looking thing that's composed of these support cells on the outside, and then packed inside of each taste bud is about 50 to 100 of these chemoreceptor cells that I've just been describing. Next slide. All right, now we're not done understanding taste yet, because at least 4 new types of tastes have been proposed and are under active investigation. We've actually found the chemoreceptor. We know it exists. We don't know exactly what it is, but it detects calcium. And we know it exists in humans because mice have it, so we went looking for it. And it interacts with a whole bunch of other organic molecules, most of which provide flavor enhancement. We also have a CO2 receptor, because the business end of the sour chemoreceptor is this enzyme, carbonic anhydrase 4. And we know it binds to CO2 because if we block it with a drug, suddenly carbonated beverages taste flat. The taste of the bubbles disappears. And that's a great way to ruin champagne, by the way. I don't know exactly what carbonated beverage they used to figure all this out, but hopefully it wasn't a good bottle of champagne. And then finally, we know that we have a taste threshold for fatty acids and lipids, as in we can't taste fat like in bacon or something like that, or in cooked meat that hasn't been defatted. But we don't know what the receptor is, but we know mice have one, and so we're looking for it. And finally, there's something called astringency, which is caused by tannins in tea, for example, and the phenolic compounds in wine. We don't know anything about that receptor. We haven't even found it in animals. But one of the things we do know is that if we cut the tongue nerve, we no longer have astringency. And then we're looking for that receptor, and then we're also looking for receptors for a whole variety of other things, like the opposite of high pH, we're looking for, or for low pH hydrogen ions, we're looking for the taste of alkaline hydroxide ions. And we're even looking for water receptor, because we know that we can't taste water until it gets to a certain threshold. And so that's sort of the first step in the discovery process. Now, I'm not going to talk a lot about the diagram on the left, but that's the signal transduction pathway for the type 2 sweet chemoreceptor. It has on the outside of the cell, this chemoreceptor cell that's inside the taste bud, it has a receptor for sugar. And that's the part that gets changed in those 25 subtypes or 40 subtypes, whatever the number was. So the sugar binds to the receptor molecule on the surface. That activates the G-coupled protein, in this case called gusducin. And it goes through a series of processes with a whole bunch of other enzymes. But the bottom line is it closes the potassium channel. That depolarizes the membrane. That opens a voltage-gated channel because of the change in voltage. And calcium ions increase inside the cell. That causes the release of the neurotransmitter, and the cell produces an electrical signal that goes to a sensory nerve. Next slide. All right. Now, we've already shown that taste is more complex and more subtle than just the 5 basic tastes, but there's a lot more to it, because we have taste buds throughout the oral cavity. In the upper part, the palate, as we start going down the throat, in the larynx, and even in the esophagus. And they provide signals as the composition of food changes, because we're chewing it. And as we chew it, those flavors, those tastes get mixed with saliva, and they change their character. And it's those taste buds which start contributing to what we call taste. Another thing that I haven't talked about is the fact that we should never pay attention to our mother's admonition to chew with our mouths closed. Because the other half of what happens, and you'll see why it's important, is as we chew, odors associated or odor molecules that can escape as we're chewing end up leaving the mouth and getting picked up by the nose. And so food actually provides additional signals that are important in what we call taste. And I'll show you that in a minute. I just wanted to show a couple of other types of receptors that produce a taste sensation. In particular, hot stuff that contains capsaicin. We call that piquancy or pungency. Capsaicin reacts with a heat receptor in the tongue. And when we get to skin, I'll show you that there are heat receptors and touch receptors and pain receptors everywhere in the body, and the tongue is no exception, although it has special ones. And in this case, it's a receptor called TRPV1. Now that receptor normally sends a nerve signal to the brain when the temperature is above about 108. What capsaicin does is it lowers the temperature when that's where that signal is sent to the brain to about 95 degrees. And so suddenly the brain is getting a heat signal that says, we're on fire. And that's the heat sensation, the burning sensation that capsaicin causes, and that's why it causes it. Now, as I said, that receptor is all over the body, including in the nose and the eyes. And so that's why if we get sprayed with pepper, our nose and our eyes burn, or we touch our eyes while we're cutting up hot peppers, God forbid, our eyes burn. It's because that capsaicin is doing the same thing that it does on the tongue in other parts of our body. Fortunately, that's a very unusual heat receptor, and it really isn't distributed like on our knee or on our arm or something like that. So we don't have to worry about pepper spray burning other parts of us until it gets to bear spray. And when the concentration of capsaicin is high enough, then suddenly, even if you inhale bear spray, which is what happened during the Capitol riots, suddenly you can have permanent injuries because there are still enough of those receptors in other parts of the body that a really high concentration in bear spray can cause serious problems. All right, and then there's another one that's the opposite, a cold receptor that reacts with things like menthol and peppermint and gives us a cool sensation in our mouth because it fools the brain into thinking that our oral temperature is colder than it actually is. And finally, there's something, that metallic taste that we get from certain substances or foods. And in fact, that metallic taste is a garnish in certain cultural foods, like in Indian foods. It's called vark, and it's produced by adding silver leaf as a garnish. We're not sure about that one. Is it a conductivity effect like you probably get from zinc? Or is it some kind of receptor effect that we are not aware of? In any event, the signals from all of these kinds of receptors reach the brain using the same nerve pathways as from the taste buds. And so all of these signals are combined in what we think of as taste. Next slide. And so let's step back for a second. Taste is the most primitive of the five senses, and it started in bacteria. But especially in higher animals, it's critical that they're able to distinguish between safe foods and harmful foods and to gauge nutritional value of foods as well. Is it worth chewing on leaf X as opposed to leaf Y, for example? And it's one of the ways that animals learn to pick their foods if they're herbivores, exactly by determining nutritional value. And so that bitter taste is important, and it has so many variants, and it's so sensitive because we can detect over 35 different plant proteins by chewing on a leaf, if you wish. And animals can really do it well. And we can taste sweet because that's fruits and honey that have high energy value. And finally, we can taste salt because that's how we determine if something has key minerals that we need. And that's why, again, herbivores are attracted to salt licks. They never get enough key minerals from their vegetarian diet. And so they're always looking for concentrated versions of those key minerals. And so some of these simple tastes, like bitterness and sweetness, require immediate reflex action. Yes, I'm going to grab as much of that stuff as I can before somebody else gets it so I can eat it. Or I'm not going to touch it because it's poison, it's too bitter. And interestingly enough, the signals from those basic tastes go straight to the brainstem. They don't go to any higher functioning part of the brain at all. Those interpretation of those signals are hardwired into the most primitive part of the brain, and we understand them without any further interpretation or contribution from higher brain activities. On the other hand, humans are able to discern over 100,000 flavors. Flavors are different than basic tastes, which is another thing that causes confusion. Flavors are a combination of all of these different kinds of sensory inputs that I was talking about. So most taste signals go through the brainstem, but then they keep going and they get distributed by the thalamus to various parts of what is called the gustatory cortex. And it's gustatory because it gets signals not just from taste, but those signals from texture, and it understands them, and signals from smells. And it combines all of those and interprets those and compares them with previous experience. I tasted that before, and I like the one prepared by this chef. I don't like the one that's supposed to be the same thing prepared by that chef. And to prove that smell and taste go together in most cases to give us a flavor, most of you, I presume, are familiar with the famous onion apple experiment. If you blindfold somebody so they can't see what they're eating, and you pinch off their nose so they can't smell, you can give them an apple or an onion and they can't tell which they're biting into. And that tells us for sure that smell, at least in that case, is a critical part of appreciating the difference between those two. Last slide, or next slide group. And I'm going to go through these fairly quickly. I don't know what time it is, but the final one that we're going to talk about very briefly is the organ of touch, and that's the skin. And as I said, it spreads out everywhere in our body, on the outside of our body, or in the orifices of our body. And it's a lot more complicated than we think it is. It's got different layers. It's got outer layers that are called glabrous, where there are no hairs, and it's got hairy skin. And each of those areas usually contains different kinds of touch receptors and often different numbers of receptors. And there are basically 7, 4 for touch, true touch, and those are mechanoreceptors. And then there's 3 kinds of neuroreceptors, 1 for pain and 2 for temperature. And I showed you that those 2 temperature ones were probably on the tongue as well. Next slide. So here are all the different kinds of touch receptors. And the 4 at the top are the ones in the skin that are true touch receptors. And some of them, like the Ruffini endings, are everywhere in the skin. Some of them, like the Meissner corpuscle, is mostly in the hairless part. So it's really dense in fingertips, which is we don't want the hairs to interfere. So it's a rapid response, and it can detect very light touches and very, very minor or slow vibrations. And that's important because we want that sensitivity in our fingers. And so the top 2 kinds of touch are both dense in the fingertips in particular. Free nerve endings for pain and temperature are everywhere. And they're not just in the outer parts of our skin, they're in skeletal muscles and in internal organs. And so they play a role in detecting pain every place. I have a pain in my chest, and that's why we know it's in our chest. And then there are things like the Pacinian corpuscles. That's a touch one. And look where it is. It's concentrated primarily in the genitals, and in women it's in breast, but it's also in men's breast tissue. Guess why it's there? It's a rapid response to pressure or fast vibrations. I won't go any further. Next slide. Oh, I wanted to stop for a second, Lou. I wanted to point out the Krauss end bulbs. They're the ones that detect cold, and they're very localized. They're only in the tongue and the lips, but they're also in the genitals. And so you know about ice cubes and their sexual connotation and use. And that's why. And so all of a sudden, knowing about these various kinds of touch receptors gives us some ideas about all kinds of human processes that we undergo. Next slide. Here's what all of those receptors look like. There's an actual picture, and then there's a diagram that shows the various parts of them. And I'm not going to go into any of it, but if you count them all up, you end up with the 7 that I was talking about. Next slide. And finally, and surprisingly for most people, in the middle of 2019, suddenly there was a report of a new organ, a new touch organ for pain. And it was this tangle of nerves that exist just below the surface of the skin. And you can see a photomicrograph of it on the right, and it's been color enhanced so you can see the various parts of that network, like the various nexuses that exist. And as of the time that I prepared the slide, we didn't know about it in humans, but we found out that it's in humans too, and it's responsible for what some people suffer from called persistent pain. And that's why some people have persistent pain. that particular pain organ goes haywire, and it just keeps firing, usually because of an injury, like the phantom pain from amputations, or the pain that we get from pulling a fingernail. It never goes away. It's there forever. And this apparently is the pain organ that's responsible. Next slide. All right, these are the processes by which signal transduction is produced. And because there are all of these different kinds of receptors, there are different processes. And we understand the ones associated with pure touch pretty well. And I'm not going to go into them. But then when we get to the nociceptors, the ones for heat and pain, we know a little bit about the heat one and the cold one. Those are the TRPV1 and TRPN8 that I talked about. And we know that those receptors, for example, are on the tongue. But the details of the signal transduction mechanisms are unknown. And even more so in the case of pain, we think we know one of the proteins involved in the process. But as you saw in some of the other pictures, some of the G proteins have all sorts of coupled cofactors that are responsible for the rest of the process. And in most cases, including even in the heat and cold ones, we really don't know what those are for sure. And so that's an active area of research, even at this late date. Next slide. Now, the other thing that's unusual about touch is because touch sends signals from all over the body, it's very different from the other senses. They're all pretty much confined to the head. And so there are very specific short pathways that go from the sense organ straight to the brain. In the case of receptors in the skin, like at the bottom of the foot, there's a very long pathway to the brain. It's very far away. And so these are really not sensory sensations that we get from touch. They're called somatosensory sensations. And that's because they use what's called the somatic nerve pathway to get the signal that's produced by one of their receptors to the part of the brain that's important for understanding what the signal means. Most of these receptors produce signals that are critical, so they all go through the medulla oblongata, part of the brainstem, very primitive. And so pain, jerk your hand away as an example. That's why that happens. But it's a very complicated process and a very important one, so I'm not going to really go into it more now. Next slide. So the takeaway lessons for the other four senses in particular are sensory receptors collect information. That information gets transformed through signal processing into a nerve signal. The nerve signal goes to the brain, and the brain makes sense of the signals that it gets. If you don't remember anything else, it's those four things. And then that will lead you back to the idea of, okay, what's the receptor that's responsible for generating the signal? And you can work your way backwards in your memory to sort of recall all of the other stuff we talked about. And I think this is the final slide next, Lou. All right, so this is a preview of the next part of the talk as far as I've thought about it so far. I'm going to talk about internal perception, and that's called interoception, and it's how we perceive ourselves. And it's a relatively new field, and the part that I'm going to talk about is the physiological part that refers to signals sent to and from the brain about the body. And in particular, I'm going to talk about proprioception, which is a part of interoception and is often called the sixth sense. And in my mind is really the sixth sense. And it's how the movement portion of interoception works. You can see up there how the body responds to external stimuli. Why and how do we know to pull our arm away from a hot stove? Or how do we know to jump off of something sharp if we step on it? How do we restore our balance without thinking about it? How do we know we should run if there's danger? All of that is proprioception at work. It's a non-conscious, for the most part, process. And it has proprioceptors, special types of these somatosensory receptors that are only found in skeletal muscles, tendons, and joints. And muscle spindles for the muscles, tendon organs for the tendons, and joint receptors for the joints. Now, the other thing is that the sense of touch is an integral part of proprioception. And so I'm going to go into much more detail about all of those skin receptors when I talk about proprioception in part 2. And now I'm done, and I hope I haven't spent too much time. Well, that's terrible. No, hour and a half. That's a little bit longer than I was planning on, but better than I usually do. All right, so at this point, I'll open it up for questions. And I really didn't stop to let anybody ask them along the way, so people must have a lot of them, assuming people are still around.
Is there a test?
No, there's no test, Lynn.
Essay questions, nothing?
I'll be happy to give quizzes if that's what people want, but there was an awful lot of information about something that people generally think they already understand. And especially when you look at something like the ear, people understand the outer ear and they know about the middle ear, but they really don't know anything about that inner ear and what goes on in the cochlea, which is really the heart of the hearing process. And when I started preparing the slides for this first version of the talk a couple of years ago, I found surprise after surprise after surprise, because the way I learn about a subject is to look at the subject and ask, okay, here's a question I want to know the answer to. And then I use Google and find the answer and try to understand it. And sometimes it's a very simple explanation, but sometimes it's a very complex piece of information that's buried in a very science discipline specific literature paper with a whole bunch of words I've never seen before and a whole bunch of concepts I know nothing about, like nerve anatomy when I was talking about interoception. I thought I knew about it, the spinal cord and the way things go up the spinal cord to the brain, but who knew that there were all of these different segmented pathways that are all separate, and the spinal cord keeps them all organized. And so that's the kind of thing I learned about, and that's the kind of thing that's important to understanding how skin receptors work, because they use a somatosensory pathway, a special set of nerves that goes through the spinal cord to the brain. And hopefully I'll be able to give a simplified version of that nerve anatomy lesson next time if I have the energy to prepare the next part. Another slide, or another talk rather. I'm slide happy, sorry. Anybody else? Yes.
I have a question. Steve, I came in half an hour late, so I don't know if you already answered this, but when did, I mean, have you studied this for a long time, or is this a recent bit of research for you, or?
And the answer is, as I said, that in certain cases like vision, I started getting interested in vision and followed the process of the research along because I was involved in understanding the very first step in that vision process from a chemical point of view. And that was that cis-trans isomerization that you probably missed when I talked about vision and the various parts of it. And so I got involved in that one back in the early 60s. I got involved in smell in the 90s or in the early 2000s when I heard the lecture by the Nobel Prize winner, Lynn. And the other ones I learned about a little bit at a time, but really most of the research for the other senses I did in preparation for the first version of this talk. And I've since done a lot more. And even after I prepared the slides, I don't know if you can see it, but I have this piece of paper with all kinds of new questions in reviewing my slide that came to mind, and I was frantically answering by doing Google searches in the half hour before the meeting started. And so it's a never-ending process for me. There's always another question around the corner that I can get interested in. And that's the way I'm interested and learn about most subjects. One of the things I learned in doing 50 years or more of research is how to do research on a new subject, especially because I had to do it many times as I basically changed fields. I was always a chemist until I wasn't, and became a biologist until I wasn't and got involved in medicine. And so I've always had to learn the basics, at least, of new fields and do specific research on specific kinds of research projects that I threw myself into without knowing anything about them. And a perfect example is when I went to CETUS in the early 80s, and I was suddenly the research director in a field that I knew nothing about, that was primarily based on cell biology. I hadn't even heard of it. And I was confronted by experts across the board. I think I've told the story of walking into a room at one of our first directors meetings, and there were the scientific advisory board sitting around a table, and they consisted of 12 Nobel Prize winners in the field that I was supposed to be directing research. And so I had to learn that one very quickly. And fortunately, I had a lot of really good people working for me. And before there was Google, there were experts that I could ask questions of. And I learned to do that in George Hammond's lab, doing undergraduate research. Why are we doing this kind of experiment? You know, I was the one actually doing it, but why did we design it this way? How come it's so tedious and I have to keep doing the same damn thing 50 times? And so by asking those questions, I learned how to ask the fundamental questions of doing research in a new field. Long answer to a very short but important question.
So Steve,
Thanks.
do you want to do your next one next week, or how would you like to do that
No,
next
I don't
week?
want
or
to do the next.
the-
I don't want to do the next one the next week. I put way too much.
Okay, would you?
I thought that I was going to give pretty much the same version of the talk that I had given a couple of years ago. And then when I started reviewing the slides, I got embarrassed. And so I spent the last week pretty much nonstop doing the research to revise all of the slides that I presented
Wow.
today. And I'm probably going to have to do the same thing for the next part. And so I need time to recover and do
Okay.
all of the things that I didn't do this past week. I don't know if you've seen my picture, but one of the things that's obvious is I need a haircut badly. And so that's on the agenda for tomorrow, for example. And I've got to go food shopping and a lot of pick up prescriptions, all sorts of things that I've been putting off for the last week in preparation for this talk. So I'm doubly appreciative of the fact that a lot of you stuck around and were interested enough to stick around to the end. Thank you very much.
Well,
Next.
thank you, and we can